Okay, good morning, everyone. Let's get started. Good morning and congratulations. Hopefully you have all finished assignment 10 and or D5. You don't seem that excited. Some excitement. Okay, great, wonderful. Okay, as promised or as mentioned last time, you've reached the end of the beginning. So you now have an experimental platform on which over the next 5 weeks, you will get to flex your increasing ability and creativity in the space of evolutionary robotics. You've had your nose to the grindstone for the last 10 weeks, so you may not have noticed. So allow me to invite you to pull back a little bit and have a look at what you've created. For many of you, you've created a code base that is much larger than you've created before. How many of you remember where the fitness value is read in your code? Which object, which class? I don't know about you, but I don't remember either. As a software engineer, this can be a bit of a scary transition where you get to the point of trying to develop and maintain, and now over the next few weeks, extend your code base without being able to hold the entire thing in your head. A tricky thing takes a little bit of practice. So before we talk about the final project, I want to talk a little bit about some ideas that will help you ensure that you are on and you maintain yourself on a firm foundation, because you're going to build some increasingly heavy stuff on top of that foundation. Okay, in the spirit of pulling back, you have now 3 executables or 3 separate processes that are running whenever you start up assignment 10. This does not go for the differentiable assignments, but for assignment 10, you have a program called search.py, which, surprise, surprise, searches over the space of all possible sets of synaptic weights for the quadruped, right? The quadruped is made up of a neural network controller. Inside that quadruped's controller, there's a whole bunch of synapses. Each and every one of those synapses needs a floating point value assigned to it. Which floating point value should you assign to each synapse to get your quadruped to run forward? Or maybe, as you start in on the final project, jump up and down, do the long jump, climb some stairs, climb a ladder, you name it. Right? So you're searching over the set of all possible vectors of a certain length, the number of synapses, where every element in the vector is a floating point number. For all intents and purposes, this is an infinite search space. It's not quite, because floating point values are finite, but it is a big, big, big space we're searching over. How does search.py do this? It uses a program called generate.py, which takes a genome, the vector of floating point numbers, and turns that into a phenotype. Generate.py, as you may or may not remember, writes out 3 different files. One file describes the environment of the robot. Remember, all the way back to A1, 2, and 3, you were creating environments with blocks inside of it. Generate.py is writing out the environment. It's writing out a file that describes the body of the robot. And finally, it writes out a file that describes the neural network controller of the robot. When we get back to lecture 13 in a few minutes, we're going to be talking about this genotype to phenotype mapping, the thing that takes the genetic instructions, the vector, and turns it into something that your search process is going to try and assign a fitness value to. So far, so good? Hopefully this is all familiar. Okay. So what takes all of this and turns it into a fitness value? For us in this course, it's a physical simulator. So you have simulate.py, which simulates a single robot, simulates all of its behavior, and then simulate.py watches some aspect of the behavior of this robot and spits out a single number to disk, the fitness value of that individual robot. And around and around and around we go. Yeah? Okay. Some of you have been struggling with parallel hill climber in assignment 9 onward, which is Search.py, instead of writing out one genotype and turning it into one phenotype and then one fitness value, it's writing out multiple genomes at once, sets of these, and then writing out multiple fitness values of this. Yeah. This is a little tricky depending on your laptop. This can very quickly get into a lot of files that are being written out, and it can be a little complicated. So I wanted to just pause for a moment, and for those that are still struggling with assignment 9 and 10, the reason that we've built our assignments into these 3 different processes is to try and make this as modular as possible. So if something isn't working, you can try and debug this by running these pieces separately. So if you haven't figured this out already, when you're debugging things, you can go into generate.py and make a few changes so that when you run it, you can run it at the command line. And when you run it at the command line, it'll write out the files. You can go in and inspect those files, make sure everything is written out in the way that you want it to, everything is there. And when you're ready, then you can run simulate.py, which is going to digest all of this, simulate all of this, and write out one fitness value. Oops, if I could type, there we go. Okay. So if simulate.py is working well, you know what it should do. It should be writing out a single value into a single file, which is the fitness of the thing that was just generated by generate. Yeah? Then you can do it again, and again, and again, and again. So you can ignore search.py if you need to debug 9 and 10, and do all of this at the command line and debug each of these pieces one at a time. Questions about any of that? Okay, so we've got these 3 processes. They are communicating with one another. They're communicating with one another by writing out files to disk. This is a horrible way to get processes to communicate with one another. There are many, many more elegant ways to enable multi-process communication on a computer. We are not using those methods in this class because they're a little bit more complicated. Some of you are already at the edge of being overwhelmed with the amount of coding in this class, so we're trying to keep things to a minimum. So the simplest way to get processes to communicate with one another is just write files to disk, and then the other one waits until it sees that file, reads it in, deletes it, and around you go. Pro, very, very simple. The con, or the cost of doing this way, is every once in a while these 3 processes will stumble because one of them will be just finishing writing up a file when the other one sees that it's there and starts to try and read it in, and the 2 processes get into trouble and they crash. Some of you may have seen this already. If you're running your parallel hill climber, your genetic algorithm for a few generations, it might work for 3 minutes, 4 minutes, 5 minutes, and then crashes. It's because there's a bit of some confusion between these processes. That's the cost. If that's becoming a big problem for you, you can read up on how to get processes to communicate with one another without relying on files. If that's something you want to tackle in your final project, you're welcome to. The other way around this problem is to make some changes to search.py so that when search.py starts up, it doesn't always just by default create random genomes. It looks to see if there are any genomes written to disk and reads them in. This is known as checkpointing. So you can alter your search process so that at the end of every generation, it stores all of the genomes in some file. And then when search.py starts up, it looks to see if there are a bunch of genomes stored somewhere. And if they are, it will start from that point. It'll start from that checkpoint. If it doesn't see a bunch of genomes written to disk, search.py will do what it usually does, which is just create an initial random population of genomes. Okay, a couple of tips and tricks to help smooth things out for assignment 9 and 10. Any questions about any of that? All good? Okay, all right, so on to the final project. as mentioned, we're going to talk about the final project in a moment, and we're going to talk about how you are going to spend the coming week between now and next Monday night, breaking your idea down into 4 bite-sized chunks, which are going to become 4 weekly milestones. The first of the 4 weekly milestones is due, surprise, surprise, next Monday at 11:59 p.m. Okay, let's talk about how this is going to go. Okay, so undergrads and graduates, you can pursue the final project in different ways. For the undergrads, you may choose a pre-formulated project from a list. So if you go back to Ludobots, and you go to Module O here, the final project, you'll see that in the final project, there are a whole bunch of sections. Those sections are listed over here. There's a whole bunch of projects with cute little tongue-in-cheek names associated with them. For example, Up, Up, and Away is evolving a robot to jump. Marching to the Beat, evolving a robot to exhibit a particular legged gait. We spent a lot of time talking about different gaits when we talked about legged locomotion. Stretch those legs. Brainiac, making a larger brain for your robot. Strength in numbers. Instead of simulating a single robot, can you simulate a swarm of robots simultaneously? And so on and so forth. Bunch of project ideas for you. If you found the 10 assignments somewhat challenging, I would recommend choosing a pre-formulated project and probably one of the easier ones. You can see all of them are listed as either easy or hard. Okay. If you found A1 through A10 relatively easygoing, or D1 through D5 easygoing, you're welcome to devise a project of your own. Regardless of whether you're taking this class for undergraduate or graduate credit, if you do devise a project of your own, talk to me and the TA, or the TA tomorrow or Monday. Make sure that it's something that we believe is reasonable within the next 5 weeks or so of the semester. You've all become evolutionary robotics experts, but you might not be able to calibrate or estimate how easy or hard the idea is that you're tackling. We will try and help you calibrate your expectations. Okay. If you choose your own idea, it must involve modifying the code base that you've been developing in this class. We want you to use what you've developed. There are other physics engines out there. There are other search methods other than evolutionary search. Please use what you've been working on as your foundation for this class. Okay. There are also for those of you that made your way through D1 through D5, there are also some final project ideas using the differentiable assignment code base, and the link is there. Okay. Not 5 weeks. There are 6 weeks remaining in the semester. So you're going to implement this final project, and you're going to implement it incrementally. One of the biggest failure modes for most students who've taken this class between now and the end of the semester is to tackle something that seems really cool, interesting, challenging, you're excited about. But those students did not break it down into bite-sized chunks. They dove in, and after 3 or 4 and then 5 weeks of the semester, it became clear that they were not going to be able to finish any part of what they had in mind. So please be sure to think carefully, not just about what you want to do, but how to break it down into 4 bite-sized chunks. Okay. I think I already said this. If you struggled with the assignments, choose an easy path. Otherwise, choose something else. Please talk to us if you're going to do something on your own. Okay. All right. Here we go. So once you have an idea, I want to make a robot that climbs stairs. I want to make a robot that kicks a soccer ball into a goal. I want to make a swarm of robots where those robots carry a big object together. Whatever your project is, once you have that idea, then think about how to break it down into a series of incremental steps, into a bunch of weekly milestones. And I want you to think about how you're going to demonstrate to the TA or me that you implemented that milestone correctly at the end of each week. Yeah? The TA and I cannot scroll through 59 different code bases every week. We haven't been doing that over the last 10 weeks, and we're not about to start that now. It is up to you to think carefully about how you can demonstrate in a few images or a 5 or 10 second video that you successfully implemented that milestone. So there's a worked example here in item 3 to show you how this might work. So let's imagine a hypothetical student who chooses to implement the up, up, and away project to make the robot jump. So you've got your quadruped sitting on the ground. You're going to try and get it to jump as high and stay in the air for as long as possible. Sounds like a great final project. This, in order to make this happen, the student has thought to themselves, it's not mentioned here, that they're going to alter the fitness function. At the moment, in your code base, F is a float that represents how far the robot travels in the X or Y direction. I forget which one it is. This hypothetical student is thinking that if they want a robot that stays off the ground for as long as possible, maybe a good way to quantify a jump, turn it into a fitness function, is to maximize the number of time steps in which the touch sensors in the feet are not firing. You may or may not remember that each touch sensor at every time step of the simulation returns either a +1, the foot's on the ground, or -1, the foot is off the ground. So how about we create a fitness function that just sums up all the negative ones. We want to maximize the number of negative ones. Okay, so the student thinks, if I'm going to do that, what I'm going to have to do is I'm going to have simulate.py write out all of the touch sensor values. So the student proposes in the first week that they're going to modify simulate.py so that when simulate.py ends, it's going to write out all the touch sensor values in a binary matrix into a file that computes all of the touch sensor values for all time steps, rather than writing out the distance the robot traveled. So the student in the first week is going to tackle altering the fitness function of this process. Okay, the student proposes to include a screenshot at the end of week 1 of simulate.py running, and then the student is going to open up a file and show that there's a whole big binary matrix in there. That's the proof they're proposing to us that they've successfully implemented their first milestone. At the end of the first week, they're then going to submit that to Brightspace, and if we see that actual short video, correct. Yeah, that's the idea. Make sense? Pretty straightforward? Okay. Okay, in the second week, the student proposes to modify search.py so that it can now read in this binary matrix and compute F, or boil F down to the total number of -1s. If the robot stays on the ground for every time, keeps all feet on the ground for all time steps of the simulation, there are no -1s. So F is going to be 0. If one of the feet comes off the ground for one time step and then goes back onto the ground, there's going to be only one -1 value in that binary matrix. F is going to be 1, and so on. Yeah. Okay. So same thing. They then propose that if they do this, if search.py reads this in, they don't need to make any other changes to their code base. One of the challenging things about the final project is thinking up what you want to do. I'm going to have a robot climb a ladder, but then where in this beast do you make changes to make that happen? We were just talking a few minutes ago about how you probably can't hold all of this in your head. I know I can't. So I want you to spend some time this week when you think of your idea, think about how to break it up into 4 pieces, and where in this process do you need to make changes to make it happen? Yeah? Okay. All right, so the student mentions that if they make this change, that should be it. Now that they've altered simulate.py to write out a binary matrix, and they've altered search.py to read in a binary matrix, they should be able to run search.py in the second week. You'll notice that in the first week, they've turned off the evolutionary process. They're not even really running their code base. They're running a small piece of it to demonstrate the change that they're making. We don't expect that every week you have your entire code base working. It's like working on a car. You put it up on the, whatever that thing is called. The car is inactive. It's turned off. Everything in the car is turned off. You're turning on bits of your code, altering them, running them, and demonstrating to us that that part is working. And then at some point, you're going to turn all the lights back on. In the case of this hypothetical student, they're doing that in the second milestone, and they're predicting that this will lead to the evolution of robots that keep as many feet off the ground as possible, which I will show with a video. So they're proposing that at the end of the second week, they're going to submit a short video showing a robot that keeps as many feet off the ground as possible. Yeah? Seems reasonable to me. It will seem reasonable to the TA. Either myself or the TA would be able to watch this video within 5 seconds, know whether this modification and this modification is causing what the student predicted would happen. So far, so good. You'll notice that the student, this hypothetical student, is quite observant and prescient. This hypothetical student did not say, the robot will jump. Why not? Keep as many feet off the ground as possible. That sounds like jumping. What do you think would happen if you, and you can try this yourself, if you modify your code base to modify the fitness function to sum the number of -1 firings of touch sensors? What are you going to get? What do you think evolution's going to find?
Some perverse instantiation where the robot rolls on its back or like bends its legs the wrong way or...
Got it. You've absorbed one of the main lessons from this course. Probably this isn't going to work. Now, it's not exactly clear how it's not going to work, but as you just mentioned, there's many ways in which the quadruped can probably embody, perversely, this fitness function. Flip on its back, dance in place, walk. None of this particular change does not specifically select for this. Yeah? That's okay. The student came up with the simplest possible change. Let's just minimize touch sensor firings, and we'll see, whatever we see, we should see the minimum of touch sensor firing, those feet off the ground for as many time steps as possible. Okay. Maybe I already gave it away here. How do you select for jumping? Summing lack of firing of the touch sensors ain't going to do it.
Change the z value?
We could change the z value, so we could incorporate the z value, the height of the robot, into the fitness function. How?
If it increases.
Okay, so maximize height. I'm the quadruped. I'm maximizing height. Part of the solution, but not enough. Tricky, right? Jumping doesn't seem that hard. There's lots of perverse instantiations of attempts to jump. What else? How might you modify the fitness function to get this? Stay up and off the ground for as long as possible. Consecutive time steps in which none of the touch sensors are firing. So the student here proposes in third week, I will modify the fitness function to compute the number of consecutive time steps that the robot keeps all the feet off the ground. You might still get flipping over, so we might need to add a penalty term for rotation of the body. Not clear. I will demonstrate this with a video in which the robot jumps and stays in the air as long as possible. Okay, so the student is guessing there might be some form of perverse instantiation. They've got an idea to deal with it in the third week. Okay. The fourth and final milestone, you're going to be moving from the alterations you've made. So hopefully by the end of week 3, you've got the rudiments of whatever you want. Stair climbing, ladder climbing, swarm behavior. May not be perfect, but you're on your way. What we want you to do in the 4th milestone and into the final project is to perform A/B testing. How many folks have heard of A/B testing before? Couple people, but not many. Okay. Whenever you go to a website, from time to time, without you realizing it, you're landing on version A of that website. The web developers for that company that make that website, they have 2 different versions of the website in mind, and they're not quite sure which one is going to convince you to spend more money. So they create 2 slightly different websites, and every time a new user lands on the landing page, the code flips a coin. Heads, you see website A. Tails, you see website B. Assuming it's a big enough company and they get enough traffic, they get thousands of data points of people using site A and thousands of data points of users using website B. And eventually, the web developers will throw away one of these versions, and either A or B will become the website. How do they decide? Whichever got the users to spend more money. That's A/B testing. It happens everywhere. It's a simple trick. Obviously, the intuition behind it makes sense. You're going to be doing some A/B testing in your final project as well. You're going to create a version. Once you get to week 4, you'll have a modified code base that's producing kind of okay jumping or kind of okay swarm behavior. And then you're going to make a slight modification, further modification, to make version B. You want to know which of 2 slightly different versions of your code base will do a better job of getting the robot to do whatever you want it to do. In the case of this hypothetical student, it's better jumping. Stay up and off the ground for as long as possible. So here's the student proposing their A/B test. I will modify simulate.py again to write out the robot's horizontal distance along with its touch sensor data. And now they're proposing a fitness function of d times t, the horizontal distance traveled by the robot, and the time steps, all feet are off the ground. They predict that this will evolve long jumping. So they're now proposing B, version B of this, which is going to evolve for long jumping, and A, which they had the week before, is jumping in place, or had said nothing about D. So maybe this process evolves this, maybe it evolves this, maybe it evolves this. It's not clear. They've got 2 slightly different versions of their code that have slightly different fitness functions. This fitness function is F equals T, the consecutive time steps off the ground. Version B is F equals DT. Okay. They're then going to compare whether evolving a standing jump, which they had at the end of week 3, or a long jump causes the robot to jump as high as possible by comparing the maximum y coordinate, I guess that should be the z coordinate, reached by the robot. They're trying to get the robot to jump as much as possible. So which of these 2 fitness functions will do the trick? Okay. Okay, that's the end of week 4, and they're going to demonstrate, sorry, I should probably add a sentence here. They need to tell us what they're going to submit at the end of week 4. They're going to submit a short video of the robot evolved to do this. So at the end of week 4, they now have version B of their code, and they now have version A of their code. Between the end of week 4 and our exam period, which is 2 weeks later, during those 2 weeks on your laptop, you're going to run A overnight. The next night, you're going to run B, then A, then B. You're going to run as many runs, as many independent evolutionary runs of A and B as you can. And then when we meet here on when we meet here on Tuesday, May 6th at 7:30 in the morning. My apologies. When you meet here, we are all going to do very short oral demonstrations where you're going to show us 2 videos of the robot evolved for T and the robot evolved for DT, and we'll see, what did your A/B test yield? Did you get better, higher, longer jumping in the case of A or in the case of B? You'll be presenting on May 6th the results of your A/B test. Okay, that's a lot of information I just threw at you. It's all in the document here. Questions? Comments?
I might have missed this in the beginning.
It's all right.
Is there an additional report that is written for it?
There is a written report as well, which we'll talk about in a moment. Clear on what you're trying to do between now and the end of the semester.
Are grad students expected to work in Tai Chi or?
Yeah, good question. Sorry, I forgot to mention. For the grad students or for those that took this course for graduate credit, you can either use A10 or D5 as your starting point. It's up to you. Okay. Let's see. Actually, on that topic, I showed you, and if you go back to lecture 1, you can watch the Robot Olympics video again. This is an example of final projects, particularly successful final projects that were conducted from the end of A10. There is in the final project, I mentioned this already, there's links to final project ideas and demonstrations of things students have tackled in previous years, continuing on from the end of D5. Yeah. Other questions, comments? Okay. Okay, so let's talk through the timeline. Okay, we are currently at March 25. So next Monday by 11:59 p.m., you're going to complete the housekeeping section in the final project Reddit page. So if we go back to the Reddit page, final project, you'll notice there's a housekeeping section here. You've been building a lot of code. There's a couple of wobbly bits that you're going to have to go back and clean up and fix up, so that when you really start cranking and doing some evolution for hours and hours on end, everything will work okay. So make sure you finish up the housekeeping first, and then complete your final milestone. You will see that in Brightspace, there is now a submission link for milestone 1. I want you to, in milestone 1, you're going to copy and paste the text descriptions of your final project, your selected project, what did you choose, and a brief description of these 4 milestones. So our hypothetical student here, next Monday at 11:59 p.m., they've written all this out and they copy and paste it into their Brightspace submission. So next Monday, you're telling me and the TA what you have in mind, and a few sentences about how you plan to break this down into bite-sized chunks. Okay. Most of you are probably now worrying, what happens if what you propose doesn't end up happening? That's okay. No problem. We want you to think ahead and plan ahead, but you don't really know what evolution is going to give you back. So in subsequent submissions, you're always copying and pasting this text in, but you can annotate this text from week to week. What do I mean by that? You can say, here at the end of week 2, here's what I promised for week 2. This didn't work, and I had to come up with a different solution. So here's the modification I needed to make this past week to the second milestone, and here's the video demonstrating that I did it. It's okay to make changes in your plan as you go, but neither the TA nor I can remember everybody's plans, so keep pasting it into your Brightspace submission. Here's my plan, here's what I proposed, here's the changes to that plan that I needed to make this week. Yeah? If you submit a wonderful video and we don't have sufficient context to be able to understand what you've implemented, how can we assess whether you've implemented it correctly or not? So make sure that you help us help you. Tell us what you had planned, what you changed, and what we're supposed to be seeing in the video, and how that observable in the video or those screenshots prove that you've made progress towards your final project. Okay. So at the end of next week, you're telling us what you have planned. You're going to summarize your 4 bite-sized chunks, and you're submitting your first bite-sized chunk, images or videos of milestone 1 in action. Okay. You don't need to include proof that you completed the housekeeping section. That's on you. Okay. And then rinse and repeat. 2 weeks from now, you're going to complete your second milestone. There will be a link to milestone 2 in Brightspace. You'll copy and paste the text descriptions of your project, your 4 milestones, and any changes you needed to make, and again, a link to proof of completion of milestone 2. Same for 3 weeks from now. 4 weeks from now, you're submitting preliminary evidence of your A/B testing, your 4th and final milestone. Okay. 5 weeks from now, 6 weeks from now, you have 2 weeks between the submission of your preliminary A/B test. You have 2 weeks to do your A/B test, collect as much A data and collect as much B data as you can. And the night before our final exam period, you're going to be submitting 2 things to Brightspace, a written report and an oral presentation. This is going to be a short video, which we'll talk about in a moment. You are then going to, the following morning, when you give your short oral presentation, I'm going to play all the videos. You will come up to the front of the room and you will talk over your short silent video, demonstrating what you implemented, showing us some of those short milestones, and showing us in the video the results of your A/B test. Good? Okay. I know it sounds like a lot, but this is distributed out over 6 weeks. The grades are distributed also over the next 6 weeks, so all of what I've just described comprises 30% of your final grade. The 4 weekly milestones, each one is worth 4%, so that's 16% of the 30%. And then the remaining 14% is for your oral presentation and your written report, which you'll be delivering and talking about in the final exam period. We will talk more about exactly how that oral presentation and written report, how you will prepare those 2 documents later. It's a lot for today. I just want to make sure you're clear on what's happening over the next 4 weeks. Okay, questions, comments? How many people already have ideas? Couple people. Okay, great. Okay, all right, that was a lot to throw at you. Let's reorient. Where are we and where are we going? We are working our way now through open problems in the field. We're going to start each of these lectures in this section with a particular problem and the best solution that bright minds in the field of evolutionary robotics have been able to come up with so far. None of them are satisfying answers, in my opinion. Maybe some of you will come up with good answers to these problems. Okay. There are 3 sort of groups of problems we're looking at. There's the genotype to phenotype map. It turns out that in an evolutionary algorithm, the way that you take genetic instructions and turn it into a solution can make it easier or harder for the search process to evolve a solution to your given problem. That's known as the genotype to phenotype mapping problem. That's something that you as the programmer have to sit down and create, and unbeknownst to you, you might create a bad mapping or a good mapping. So we're going to dive back into lecture 13 in a moment, where we started to talk about the NEAT algorithm. The NEAT algorithm is an attempt to create a good genotype to phenotype mapping, where a good mapping is something that makes it relatively easy for the evolutionary algorithm to evolve a solution to your problem. We'll finish that today, and we will move on to one of the more fun and scary parts of the course, crossing the reality gap. Super hard problem. No one solved this very well yet. We're going to look at 4 different solutions to the reality gap problem. There are dozens of solutions out there in the literature. These are some of the more famous and well-known solutions. Okay. We'll end this section in a few weeks by looking at scalability. How do we get more and more people to evolve more and more robots to do more and more things? And how do we also manufacture more and more numbers of physical instances of those evolved machines? If evolved robots are actually going to be useful here in the real world, we're going to have to design, manufacture, and deploy very large numbers of them. No one knows how to do that yet, but there are ideas. Okay, so back to lecture 13, neat and hyper neat. Just as a very quick reminder, we're dealing with the genotype to phenotype mapping problem. And I showed you another problem last time, the competing conventions problem, that demonstrates why arbitrarily choosing a genotype to phenotype map can often be a bad idea. If we're evolving neural networks to compute exclusive OR, and we use a simple mapping, meaning we take the genomes, the two sets of weights for these two neural networks, and we cut them at random and glue the two bits together to make a new child neural network. That doesn't work very well because of the competing conventions problem. If you don't remember all the details of the competing conventions problem, that's okay. It was just meant as an example of doing things simply, like evolving sets of synaptic weights, grabbing two sets of weights, grabbing two parents, cutting them and gluing the parts together, makes things very hard on the search process. You can run your search.py for hours, days, months, years, and decades, and you might never be able to evolve a neural network that solves the simple exclusive OR problem. So Stanley and Mikulainen, many years ago now, came up with the NEAT algorithm, which is a way to evolve sets of weights, ways to evolve neural networks so that you can cut them and recombine them and get neural networks that are better than the parents. How did they do this? They started in on the NEAT mapping by altering G first. Then we're going to see how they turn G into P. This is where we ended last time. G is no longer a vector of floats. G is now a more complicated data structure. It's a pair of lists. The first list includes information about the number of nodes or the number of neurons in the neural network. So here's the phenotype down here. Here's the neural network. Here's its physiology or neural architecture. It's made up of 1, 2, 3, 4, 5 neurons because the genome said add 1, 2, 3, 4, 5 neurons. Inside each element of this list, it also indicates which kind of neuron to add. So add 3 sensor neurons, 1, 2, 3, add a hidden neuron, and then add an output neuron at the end. The second list dictates how to wire up all of these neurons. Connect neuron 1 to 4 with a synapse that has a weight of 0.7, then add a second synapse that connects neuron 2 to 4, give it a weight of minus 0.5, and so on. So as we move along this list from left to right, we wire up all of the neurons. And then inside each element of this second list, there is some additional information. You'll notice this fourth row here, enabled or disabled. This is a binary number inside the genome that indicates whether that synapse is enabled or disabled. This seems like kind of an odd thing. So you'll notice there's a synapse that is supposed to connect neuron 2 to neuron 5, but in the phenotype, there is no such synapse. It's been disabled. It's been turned off. They're, in essence, building in commenting and uncommenting to the genome. In your code base, I'm going to guess that there are many lines of code that are now commented out. And some of the things you probably do is uncomment and then recomment parts of the code base. One of the more interesting things in the early days of genetics was the discovery that not every part of your genome codes for some part of your phenotype. Many parts of your genome, and it turns out quite a bit of it, is actually commented out. Kind of odd. Turns out, and we don't have time to get into this, there's actually good evolutionary reasons for doing this. Why do you do it? Why do you comment in and out parts of your code?
Is it like protection against mutation?
If a mutation hits this particular synapse, if a mutation lands right here and changes the weight from 0.5 to something else in that child is going to behave, is going to have an identical phenotype to the parent. So one of the reasons that inoperative code might exist in us is because it buffers against mutation. There are other reasons why it might be useful. We don't have time to go into it now, but it's in there. The fifth and final row, there's this cryptic term here, innov, which is short for innovation number. The innovation number is the heart of NEAT. It's why NEAT works, and it's why 24 years later, depending on what you're doing, people still use this algorithm to evolve neural networks. Okay, we're going to come back to the innovation number in a moment. Let's have a look at some other aspects of NEAT for a moment. Here are here's one parent up here. Here's the phenotype. This particular parent has 1, 2, 3, 4, 5 neurons and 1, 2, 3, 4 synapses in it. And here is now just the second list. So from now on, I'm not going to draw the first list, which specifies neurons. I'm just going to show the list of synapses. How do you know this is a synapse list? Because I'm going to use this shorthand. Synapse 1 wires neuron 1 to 4. Synapse 3, down here, synapse 3 wires neuron 2 to 4, and this particular synapse that wires neuron 2 to 5 has been disabled. No connection here. These numbers at the top here, these are the innovation numbers, which we haven't talked about yet. We'll come back to the innovation numbers in a moment. Okay. As we're turning the genotype into the phenotype in NEAT, there are a number of variation operators. There are a number of ways in which mutation and crossover can vary a child's G relative to its parent's G. We can mutate G. So if this parent survives and produces an offspring, we copy all of this genetic information, and as we're copying it, we make a random mistake. One of those random mistakes is to add a new synapse. This we haven't actually seen before. In the code that you're running right now, your G, your vector of floating point numbers, that vector never gets longer or shorter. You never add, or mutation never adds or removes synapses. Mutations only alter the weights of a set number of synapses. So now we have a genotype to phenotype mapping, which can lengthen or shorten the genome, which has an effect on the phenotype. We can see that in this case, a mutation added a new synapse to the end of the child's G, and that new random synapse happens to connect by chance neuron 3 to 4 with some random weight. And again, I'm not writing out the weights anymore. Okay, so we can add a connection or add a synapse. Alternatively, a mutation can take, again, this parent, and instead of adding a synapse, it randomly adds a node. So in this case, mutation has added a new node or a new neuron 6. 6 is hanging out there in space. It's not connected to anything. So some additional mutations eventually connect 3 to 6 and 6 to 5. So mutations can add neurons and synapses, and although it's not shown on this slide, mutations can also delete neurons and synapses as well. And we also have the more familiar types of mutations like change the synaptic weight of an individual synapse and so on. So the number of ways in which the evolutionary algorithm can alter G is increasing. Modify, add, delete. Yeah, a bunch of different mutation operators. Okay, now we come to the most fun part of NEAT, where we're going to need these innovation numbers. We're going to attempt crossover. We've got parent 1 here, parent 2 here, and this may be difficult to see from the back of the room. They're slightly different. Their phenotypes are different. They have different structure, and they have different genotypes. So in parent 1, there are 1, 2, 3, 4 synapses. 1, 2, 3, 4 synapses. In parent 2 over here, there is 1, 2, 3, 4, 5. 1, 2, 3, 4, 5 synapses. And also, oh, by the way, parent 1 and parent 2 have a couple of disabled synapses. Their G's are different and their P's are different. How are we going to cut these parents and glue the two parent parts together to create a new child so that that child looks kind of like its parent, kind of looks like its parents. So what do I mean by look like? Meaning it has more or less the same neural architecture. And how do we ensure that this child neural network also behaves similarly to these two parent networks? This is tricky. This is the competing conventions problem. If we do this naively, if we just cut them and glue them together, it's unlikely to work. So the crossover event that we're going to observe in a moment is based on an intuition that these authors had. Let's go back to these two hypothetical neural networks for a moment. Okay. As you remember, these two neural networks exist in a population of evolving neural networks, and they're all being evolved against a fitness function that selects for how well they do exclusive OR. And in order for these neural networks to do exclusive OR, somewhere inside the body of each neural network, one of the hidden neurons has to be kind of doing and the other hidden neuron has to kind of be doing OR. And then they kind of have to be combined in the right way at the end. Let's imagine that we're partway through the evolutionary process, so it's all kinda. Some of them, each and every one of the neural networks is kind of doing and inside and kind of doing or inside and kind of combining them in more or less the right way. Let's assume that these neural networks have evolved. They're the result of a whole bunch of previous mutation and crossover events. They've come from the same set of ancestors. All of us in this room, we're all related. We may be 1,013th cousins of one another, but we do have common ancestors if you go back far enough. Looking around this room, I see some blue eyes, some green eyes, and some brown eyes. So we have some variation, but we have more or less the same trait, eye color. And it turns out that eye color actually can be localized to one part of G. And it turns out that the place on G in you, where your eye color is encoded, is the same place in me where my eye color is encoded. We might have different alleles. We might have different versions of the gene at that location in G, but it's at the same location in our genomes. Why? Why isn't my eye color located on my chromosome 3 and your eye color located on your chromosome 22?
Because when you combine two parents' genotypes, the eye color is encoded in two different places, and you can have two different eye color encodings.
So you're exactly right. So if eye color was in different places, then crossover could produce a child that has two copies of the eye color gene, maybe not too problematic, but it could also produce another offspring that has neither, has no version of eye color gene. Not a great situation. So what you just described is correct. It's the benefit of having specific traits at the same place in the genome. That's a benefit. But why is it so? Why does it happen? So I'm asking about mechanism. Where did it come from? It's good that we have it, but where did it come from?
Isn't it from like mitosis and meiosis because they line up before they split up?
Yes, you're describing mechanism. That's good. Mitosis. When it splits, it assumes that everything's in the right place. I apologize. You're having to mentally brush off first year bio, which some of you may not have taken. Eye color and all of our traits are all in the same place in our genomes because we have the same common ancestor. There was a period in human history when there was about 10,000 humans on the planet. There were periods in our past where there were very few of us. So we all came from more or less the common ancestor, and those few humans, they had eye color in one place on the genome, and we are all their descendants. So the fact that we have a common ancestor, that's the reason why. That ancestor had eye color on, I don't know which chromosome it is, it's on chromosome 5, let's say. So that individual's offspring had eye color on chromosome 5, and their offspring, and their offspring, and their offspring. So we started this lecture by saying it's very difficult to know where and is in this neural network and is in this neural network if we just look at these two neural networks. But if we walk backwards in the evolutionary history of these two neural networks, they exist in the population because their parents had good fitness. So if we go back far enough in this evolving population, we're probably going to find a common ancestor of these two neural networks. One ancestral parent, and that ancestor had and in one particular place. That ancestor might not have been very good at computing and inside, but that parent got a relatively high fitness, so it was probably starting to evolve and inside. And for the sake of argument, let's just assume that and was starting to evolve in that ancestor at hidden neuron 1. So when that parent produced 2 or 3 or 4 or 5 offspring, those offspring were able to compute and inside their neural network bodies at hidden neuron 1. So if we could trace back for every bit of these neural networks to where that bit existed in the common ancestor, we'd know. We could make a guess about the function of that bit. That's the intuition underneath the neat algorithm, and I don't want to go any further unless that's clear. It's possible that these two offspring, this offspring has and over here and over here, but as long as we can trace back to where this genetic material was inherited from, and we can trace back where this genetic material was inherited from, and if we can trace these two bits of genetic information all the way back to a common ancestor, and where in that common ancestor it happened, we can be relatively confident that this bit and this bit are computing more or less the same function. Maybe not identical, because it might have changed over time. Maybe this is green eyes and this is brown eyes, but it's still eyes. It's still doing more or less the same thing. So far, so good? Everyone get the intuition? Okay, so that's what the authors built into the NEAT algorithm. They're going to do very careful bookkeeping. For every single synapse inside every single neural network, they're going to know whether this synapse, for example, derived from, and this synapse in this child, that they came from exactly the same single synapse in an ancestor. How do they do that? They do that using this innovation number. So bear with me for a moment. Let's go back to the mutation operator here. So this particular neural network, there was a random mutation that added a new synapse to this particular neural network. Every time a mutation operator adds a new synapse or a new neuron to any neural network in the population, it's assigned a new innovation number. Inside the NEAT algorithm, there's a global variable called innovation number, which is an integer, and it just It just increments. Every time there's a new genetic bit that's added, that innovation number is assigned to that synapse or neuron, and that global variable is incremented by 1. If this child survived and produced a child of its own, and that grandchild also through mutation was given a new synapse, that new synapse would get an innovation number of 8. If some other neural network in the population produced another child that got a new neuron, that neuron would get an innovation number of 9, 10, 11, 12, 13, 14, 15. Throughout the evolution of this population of neural networks, every new bit of neural machinery gets its own unique innovation number. So far, so good? Okay, so let's continue this thought experiment. We have this child which has a new synapse called synapse 7, and synapse 7 connects neuron 3 to 4. Let's assume that this child has higher fitness than its parent, and so the parent dies out, but the child survives into the next generation. And then this child produces 2 grandchildren. During the copying process to create grandchild 1 and grandchild 2, we copy all of the genetic material from this parent into the two grandchildren. So both grandchildren have a synapse that connects 3 to 4. And in both grandchildren, that synapse has an innovation number of 7. We only increment the innovation number when mutation is adding a new bit to a genome. When we're copying genetic material from a parent into a child, we copy the innovation numbers. So now we have a parent that has a synapse with an innovation number of 7, and we have 2 children that also have 2 synapses that have innovation numbers of 7. Those 2 siblings, they have 2 bits of their phenotype. They have a synapse with probably a similar or identical weight that within each of the 2 grandchildren connects neuron 3 to neuron 4 in the same way, and it has the same innovation number. Because these 2 siblings have a bit of their genotype that has the same innovation number that came from a common ancestor, however this synapse is contributing to the behavior of this neural network, the way this neural network transforms its input into an output, synapse 7 is probably contributing in similar ways in the 2 siblings. Everybody see that? Okay, so we now know that there are 2 bits of semantically similar, 2 bits of material that are doing kind of the same thing in both siblings. This sounds a little bit gross, but if we cut the 2 siblings and glue them together, a little bit of incest here, we're going to produce a grandchild from these 2 siblings. What do we want to make sure does not happen during this crossover event? We know something about these 2 siblings. They have 2 pieces within them that are doing more or less the same thing, and they came from a recent common ancestor. What do we want to ensure when we try and genetically recombine parts of the genetic material from these 2 siblings? What do we not want to have happen? Any ideas? Did you have an idea?
Evolve backwards.
We don't want to?
Evolve backwards.
What do you mean by evolve backwards?
Since they're so similar. Never mind.
That's okay. Other ideas?
Like 7 is you want to keep it, so you want to make sure that it doesn't get turned off.
You want to
Like,
keep
that it gets carried over as well.
You want to keep it, make sure it carries over. You don't necessarily know that synapse 7 in both siblings is good. The only thing you know is that synapse 7 in both siblings is probably doing the same thing. It's probably got the same weight and doing more or less the same thing because it's a copy, it's 2 copies from the parent.
You just don't want 2 of them.
We don't want 2 of them. It's back to the eye color example. However crossover is going to happen, however you're going to try and combine the genetic material from these 2 neural networks, you want to do it in a way that you only get one copy of synapse 7. You don't want to get 2 copies of 7, and you don't want 0 copies of 7. How do we ensure that in the crossover operation? It's easy. They both have the same innovation number. There's a integer associated with them that says we're the same. We came from a genetic bit in our ancestor, and since it's a copy of that bit, it's probably doing the same thing in both of us. Maybe good, maybe bad, but just make sure there's only one version that gets into the grandchild in this case. That's the intuition. That's what the innovation number is doing. So in crossover, when crossover occurs at random in the NEAT algorithm, the crossover event is going to rely on innovation numbers. Let's see how this works. Okay, we've got parent 1 here, parent 2 here, and here's the G of parent 1, here's the P of parent 1, the G of parent 2, and the P of parent 2. As promised, I'm only showing the genetic description of the synapses. I'm not showing the genetic description of the nodes. We've got 5 synapses here. One of them is disabled. 7 synapses, 1, 2, 3, 4, 7 synapses over here. 2 of them are disabled. We're going to try and cut these 2 neural networks in half and glue them together into one offspring neural network. Here we go. We're going to take the first gene from parent 1, the description of the first synapse in parent 1, and put it here. And we're going to find that this particular synapse has an innovation number of 1. So we go looking in the list from parent 2 to see if there's any synapse in parent 2 that has an innovation number of 1. It turns out there is. So this particular synapse is an allele of this synapse. This synapse in parent 2 and this synapse in parent 1, they're a common inheritance from some ancestor of these 2 parents. So we're going to take this allele and place it right underneath the allele from parent 1. Again, if you squint and try and think back to intro bio, you are a diploid creature. You have a gene, and at every gene, there are 2 alleles, and they're sitting right next to one another. If you produce an offspring, your offspring will get, at random, 1 copy of the pair of alleles that you have. You may have brown eyes, and you may be carrying the brown eye allele, but unbeknownst to you, you may be also carrying the blue eye allele. You might give brown eyes or blue eyes to your offspring. It's similar function. It's about eye color, so you're only going to give 1 copy to your offspring. Same thing here. We've lined up the 2 alleles for synapse 1. We keep walking now from left to right along parent 1's genome. We take this synapse that has an innovation number of 2, we put it here, do the same thing. Go back to parent 2 and go looking for an allele. We find one. There is a synapse in parent 2 that has an innovation number of 2. We put it under here. There is no innovation number 3 in parent 1, but there is an innovation number of 3 in parent 2. So we found a particular synapse inside parent 2 that we put down here, and this particular synapse has no analog in parent 1. So we leave a little bit of a gap. We go on to innovation number 4, and we continue. We're taking the genetic material from both parents, and we're lining it up in this way. Question?
So, would this, in theory, protect against, like, genetic diseases?
It absolutely protects against genetic diseases in neural networks, where a genetic disease in this sense is we're trying to evolve neural networks that perform exclusive OR. And in order for a neural network to compute exclusive OR, it only needs to compute AND once inside and OR inside once. A genetic disease would be when we tend to cut parents and glue them together. A lot of the time we get offsprings that have 2 ORs inside but no AND, or 2 ANDs inside but no OR. If we don't line up alleles in this way, that's the kind of disease that happens. And the NEAT algorithm is designed to protect against those kinds of genetic diseases, which is presumably also why alleles and diploidy evolved in certain higher animal lineages, including ours. It protects against genetic diseases in the sense of increasing the probability that when you give sub-solutions to your offspring, you give them copies of genes, like eye color, your offspring gets one and only one copy of everything. Actually, it gets 2 copies, gets one from each parent, but usually one shields the other. In essence, you're getting one and only one thing from each of your parents, and not 2 of one thing and none of another thing. That's a very common genetic disease. So far, so good? Yep.
So if something like, I don't know what it would be comparable to an algorithm, like if something like sickle cell anemia popped up, would you have to artificially add it into the algorithm to prevent against it, or would it figure it out on its own?
That's a good question. I don't know what the analog would be of sickle cell anemia in particular. But if you ended up with something that had 2 ands and no ors, for example, is that what you had in mind, or something else?
I don't know, I mean, I guess it would be a completely rogue thing, and not part of the and or.
Something that's just
like
bad, that's in
yeah.
addition.
Yeah.
Then you better hope that the evolutionary algorithm just kills off that thing, and whatever the G that's encoding for sickle cell anemia, something that disrupts the computation of exclusive OR, that's what the evolutionary algorithm is for. Get rid of it. The NEAT algorithm is focusing on make sure everybody gets one and only one copy of stuff from the parents. Okay, so we're halfway through the crossover event here. We've taken the genetic material from both parents, lined it up, and now we're going to start to produce the offspring neural network. We're going to walk from left to right, and every time we come to a synapse or a bit of genetic material in which there are 2 alleles, we have 2 copies, one from each parent that have the same innovation number. Flip a coin. Heads, you get the version from parent 1. Tails, you get the version from parent 2. In this case, the coin came up tails, so we copy parent 2's allele for synapse 1, which is the one that connects neuron 1 to 4. This one here goes into the offspring. In parent 2, this particular synapse was disabled. It stays disabled in the child. We go on to the next synapse in this aligned list. Again, for synapse 2, there were 2 copies in the parent. We flip a coin. In this case, the coin comes up heads, and we copy parent 1's allele into the child. We come now to a disjoint synapse. There's a synapse that exists in parent 2, and parent 2 had a high enough fitness to survive and produce offspring, so it's probably good. So let's just copy it into the child. Flip a coin, copy, flip a coin, copy, copy. And now we've cut parent 1 and 2 and genetically recombined their genetic materials.
I'm just curious about the inclusion whenever there's a disjoint for excess.
Yep.
So it still seems to favor bloat. where
Great question.
We're not
Yes.
kind of entertaining the idea that parent 1 also survived without innovation 3.
Yeah.
And maybe that's better if we're always just including stuff.
Yes, great point. There is a little bit of an inefficiency in NEAT. It leads to what you mentioned, which is bloat. You can see it already. I think you can see it already. Yeah, so the size of offspring, the offspring's G is 8, and parent 7 had 7, parent 1 had 5. So the amount of genetics inside the offspring is bigger than the amount of genetics in either parent. That's bloat. Yeah, maybe not such a great idea if this continues. You're going to get larger and larger genomes, unless larger and larger genomes with more and more synapses hurts your fitness, in which case you're gone. Okay, good point. It might be a problem. Okay, so if you look at G here, it looks similar to the G's of parent 1, parent 2. And if you look at the phenotype of the offspring here, the neural architecture, the number and wiring up of the neurons in the offspring looks similar to both parents, but not identical. So far, so good. It looks like NEAT is working. What this slide does not show you is the behavior of these 3 neural networks. How well and how does this one compute exclusive OR? How and how well does this one compute it? How and how well does this one compute it? It doesn't show you. So next time we're going to look at some data to see, do the offspring tend to behave similarly to the parents? If they do, that suggests that this idea of keeping track of common ancestry helps in ensuring that crossover tends to bring together the best of both genetic worlds from 2 parents. Have a good rest of your day. You have a quiz due tonight, and you're thinking about ideas for your final project. See you on Thursday.